Hello, and welcome to your Microbiology Bio 203 lecture on adaptive immunity. I am Mr. Kennedy, and I will be guiding you through this material today. To begin, the first thing I'd like to do is define what adaptive immunity is. Adaptive immunity are your defenses that target a specific pathogen. They vary from your innate immune response, which are those things that you are born with and designed to defend you against all comers. Adaptive immunity is usually acquired through infection or vaccination, and it involves a primary and secondary response. The primary response is the response that we get to the first time the immune system combats a particular foreign substance. The secondary response is a response that occurs to later interactions with the same foreign substance. The primary response is often slow, but can also develop memory that will make a secondary response faster and more effective. Next, we'll consider the dual nature of adaptive immune responses by first considering humoral immunity. Humoral immunity produces antibodies that combat foreign molecules known as antigens. B cells are lymphocytes that are created and mature in our red bone marrow. B cells can recognize antigens and make antibodies. They're named for bursa of Fabricus in birds. Cellular immunity. Cellular immunity produces T lymphocytes. These recognize antigenic peptides processed by phagocytic cells and mature in your thymus. T cell receptors, or TCRs, on the T cell surface contact antigens, causing the T cells to secrete cytokines instead of antibodies. This illustration gives us an idea of how T cells and B cells are differentiated from each other. Cellular immunity attacks antigens found inside cells. Viruses and some fungus and parasites are attacked this way. Humoral immunity fights invaders outside cells. Bacteria and toxins are dealt with this way. Cytokines. Cytokines are chemical messengers produced in response to a stimulus. Cytokines include interleukins, chemokines, interferons, tumor necrosis factor, and hematopoietic cytokines. Interleukins are cytokines between leukocytes. Chemokines induce migration of leukocytes. IFNs, interferons, interfere with viral interactions of host cells. TNF, or tumor necrosis factor, is involved in the inflammation of autoimmune diseases. Hematopoietic cytokines control stem cells that develop into red and white blood cells. The overproduction of cytokines can lead to a cytokine storm. Antigens. Antigens are substances that cause the production of antibodies. They are usually components of invading microbes or foreign substances. Antibodies interact with epitopes or antigenic determinants on the antigen. Haptenes, antigens too small to provoke an immune response, can attach to carrier molecules. This is an illustration of how antigens and antibodies may interact. Antibodies are globular proteins. These globular proteins, called immunoglobulins, or Ig, come in a wide variety of forms, depending upon what's attached to them. Valence is the number of antigen binding sites on an antibody. Bivalent antibodies have 2. Four protein chains form a Y shape in each antibody's base structure. 2 identical light chains and 2 identical heavy chains are joined by disulfide links. Each antibody also has a V, or variable region, at the end of the arms that bind with epitopes. A constant region, or Fc region, is the stem, which is identical for a particular Ig class. There are 5 classes of Ig: IgG, IgM, IgA, IgD, and IgE. This is an illustration of the base structure of an antibody. IgG. IgG is a monomer, and it makes up 80% of serum antibodies. It's found in the blood, lymph, and in your intestine. It can cross the placenta, trigger complement, enhance phagocytosis, neutralize toxins and viruses, and protect the fetus. IgM is a pentamer made of 5 monomers held with a J chain. It makes up 6% of serum antibodies, remains in blood vessels, can cause clumping of cells and viruses, and is the first response to an infection, only short-lived. IgA is a monomer in serum, dimer in secretions, and it makes up 13% of serum antibodies. It's common in mucous membranes, saliva tears, and breast milk. It can prevent microbial attachment to mucous membranes. IgD is also a monomer. It makes up 0.02% of serum antibodies. Its structure is similar to IgG. It's found in blood, lymph, and on B cells. There is no well-defined function for IgD, but it is thought to assist in the immune response on B cells. IgE is a monomer making up 0.002% of serum antibodies. It can be found on mast cells, basophils, and in blood. It causes the release of histamines when bound to antigens and lysis of parasitic worms. This table is a summary of all Ig or immunoglobulin classes. Humoral immunity response process. Looking at the humoral immunity response process, we'll start with clonal selection of antibody-producing cells. Major histocompatibility complexes, or MHC genes, encode molecules on the cell surface. There are 2 classes of MHC. Class 1 MHC are on the membrane of nucleated animal cells, and they identify self. Class 2 MHC are on the surface of antigen-presenting cells, or APCs, and they include B cells. Inactive B cells contain the surface Ig that bind to antigens. B cells will internalize and process that antigen. Antigen fragments will then be displayed on MHC class 2 molecules. T helper cells, or Th, contact the displayed antigen fragment and release cytokines that activate B cells. The B cell undergoes proliferation or clonal expansion. This illustration shows us the process of activating B cells to produce antibodies. Clonal selection differentiates activated B cells into antibody-producing plasma cells and memory cells. Antibody-producing plasma cells do as their name implies and produces antibodies. Memory cells do as their name implies. They remember the foreign antigen for future reference. Clonal deletion eliminates harmful B cells. This illustration shows us clonal selection and differentiation of B cells. T-dependent antigen. In clonal selection of antibody-producing cells, we must consider first T-dependent antigens, then T-independent antigens. T-dependent antigens are antigens that require a T helper cell to produce antibodies. T-independent antigens can stimulate the B cell without the help of the T cell. The T-independent antigens will provoke a weak immune response, usually producing IgM, and no memory is generated. The T-dependent cells are the polar opposite. Antigen-antibody binding and its results. An antigen-antibody complex forms when antibodies bind to antigens. The strength of the bond is the affinity. It will protect the host by tagging foreign molecules or cells for destruction. Destruction can include agglutination, opsonization, antibody-dependent cell-mediated cytotoxicity, neutralization, or the activation of the complement system. This illustration gives us the results of antigen-antibody binding. Cellular immunity response process. In cellular immunity, cells, rather than chemicals, protect us. T cells combat intracellular pathogens. These T cells mature in the thymus. Thymic selection eliminates immature T cells, and those mature T cells will then migrate from the thymus to lymphoid tissues, attached to antigens via T cell receptors or TCRs. Pathogens entering the gastrointestinal tract will pass through microfold cells or M cells located over Peyer's patches. This transfer of antigens to lymphocytes and antigen-presenting cells initiates the immune response. Dendritic cells, or DCs, will engulf and degrade microbes and display them to T cells. These dendritic cells are found in your skin, genital tract, lymph nodes, spleen, thymus, and your blood. Macrophage may be activated by cytokines or the ingestion of antigenic material. They will migrate to the lymph tissue, presenting antigens to T cells. Classes of T cells. First, we consider clusters of differentiation, or CDs. CD4 is a T helper cell that produces cytokine signaling with B cells, can interact directly with antigens, bind to MHC class II molecules on B cells and APCs. CD8 is a cytotoxic T lymphocyte, or CTL, which will bind to MHC class I molecules. TCR on the TH cell recognize and bind to antigen fragments on the MHC class II or APCs. APCs or TH secrete a costimulatory molecule activating the TH cell. The TH cells will then produce cytokines and differentiate into TH1, 2, or TH17 cells. Some will become memory cells. This is an illustration of the activation of the CD4 T helper cell. TH17 cells can produce IL, interleukin 17, and contribute to inflammation. TH1 cells can produce IFN, which activates macrophage, enhances complement, and stimulates antibody production that promotes phagocytosis. TH2 cells can activate B cells to produce IgE and activate eosinophils. This is an illustration which gives us the lineage of effector T helper cell classes and the pathogens targeted. T regulatory cells, or Tregs, are a subset of CD4 cells, and they carry an additional CD25 molecule. T regulatory cells, or Tregs, suppress T cells against self, and they protect intestinal bacteria required for digestion and the fetus. T cytotoxic cells, CD8 T cells, are activated into cytotoxic T lymphocytes, or CTL, with the help of TH cells and costimulatory signals. CTLs recognize and kill self cells altered by infection. Self cells carry endogenous antigens on a surface presented by MHC class I molecules. CTL releases perforin and granzymes that induce apoptosis in the infected cell. This is a picture illustrating the killing of a virus-infected target cell by cytotoxic T lymphocytes. Apoptosis is programmed cell death. It prevents the spread of infectious viruses to other cells. Apoptosis cells cut their genome into fragments, causing the membranes to bulge outward via blebbing. Extracellular killing by the immune system. Natural killer cells, or NK cells, are granular leukocytes that destroy cells that don't express MHC class I self-antigens. NK cells kill virus-infected and tumor cells and attack parasites. NK cells are not always stimulated by an antigen, and they kill by forming pores in the target cell, leading to lysis or apoptosis. This is a table that illustrates the principal cells that function in the cell-mediated immune response. Antibody-dependent cell-mediated cytotoxicity. Protozoans and helminths are too large to be phagocytized. Protozoans and helminths' target cell is coated with antibodies. In your immune system, cells will attach to the Fc regions of those antibodies. The target cell is then lysed by chemicals secreted by an immune cell. This is an illustration of how a multicellular creature like a helminth or a large cell like a protozoan will be attacked by cell-mediated cytotoxicity. Immunological memory. The second memory response, occurs after the second exposure to an antigen. Typically is more rapid, lasts many days, and greater in magnitude than a primary response. Memory cells produced in response to the initial exposure are activated by the secondary exposure. Antibody titer is the relative amount of antibody in serum, and it reflects the intensity of the humoral response. IgM is first produced, followed later by IgG. This graph illustrates the primary and secondary immune responses to an antigen. Types of adaptive immunity. Natural acquired active immunity is one form of adaptive immunity resulting from infection. Natural acquired passive immunity is transplacental or via colostrum. Artificial acquired active immunity is achieved by injection of vaccination or immunization. Artificial acquired passive immunity is acquired by injection of antibodies. This graphic illustrates the types of adaptive immunity. Antisera and serology and globulins are the last things to consider in this lecture video on adaptive immunity. Antisera are blood-derived fluids containing antibodies, which can be used in an adaptive immune response. Serology is the study of reactions between antibodies and antigens, and globulins are serum proteins, serum fractions that contain antibodies that can be used to develop adaptive immunity. This concludes your adaptive immunity lecture video.